Further ado, I'd like to welcome our very first in the series, Dr. Catherine Eimers, and she's a faculty member in the Trent School of the Environment at Trent University, and that's in Peterborough, Ontario. She's been doing research in the Lake of the Woods Basin since 2018, and her team is currently monitoring rivers as well as atmospheric deposition at several sites in the Canadian portion of Lake of the Woods and its watershed to better understand the sources of nutrient delivery to the Rainy River and to Lake of the Woods. So I'd like to welcome Dr. Eimers and hand the floor over to you. Thank you so much for being here.
Thanks, Kelly. Oh, and already having issues. Sorry. Okay, so first of all, I just want to acknowledge that a lot of people have helped put together the data that I'm going to share with you today. So in particular, Andy Williams, who I believe is on the call, Wes Greenwood, they have been the two primary field leads on this project. And then Kelly McGillivray was a master's student about a year ago, and she also contributed a lot of the data that I'm sharing today. So to start off with and how I sort of became involved in Lake of the Woods research, I think this image on the right kind of tells it all. And this is an image from Environment Canada's EO Lake Watch website. And it shows a fairly large algal bloom that's affecting the lake right now. This image was from last week, I believe. And algal blooms like this continue to affect the Lake of the Woods, cause lots of public and political concern on both sides of the border, and have generated a lot of research and monitoring activity to understand the sources and controls of nutrient enrichment. We continue to believe that nutrients are a primary driver of algal blooms. However, we now understand that climate may also play a role. Warming waters are generally more stable and can cause more internal loading from the lake sediments. And so that's something that we're also looking at. And changes in land use are also of concern. In particular, I'm going to talk a little bit more about this later, the role of agriculture in the southern part of the basin. There's a lot of interest in expanding agriculture, right? And especially in the Canadian side of the watershed, where Canada's not exactly well endowed with area of land that's climatically suited to agriculture. So we're interested in sort of expanding agriculture, but this may have some unintended consequences for water quality that I want to sort of draw to your attention. So algal blooms really kind of is what draws people together and focuses interest on the Lake of the Woods. And a number of years ago, water quality concerns in the American part of the basin, or American part of the lake itself, initiated a TMDL study, or total maximum daily load study, by the MPCA, or Minnesota Pollution Control Agency. And that study is complete now, and the report was finalized and approved by the EPA. And if you look at that report, you'll see that, the report obviously focuses mainly on the American part of the basin because it's an American sort of management document, but you sort of get the impression that there's not as much monitoring data available for the Canadian portion of the basin. And there's a variety of reasons that sort of contribute to that. For one, it's a very large basin. It accounts for, almost 2/3 of the total watershed draining into the Lake of the Woods is in Canada. It's rather remote. We don't have any major cities in the basin, sort of base monitoring efforts from. And so we don't have as much information within the Canadian portion of the basin to guide management. And that's kind of where I came in. And so 3 years ago now, I was approached initially by somebody from the Ministry of Environment, and then put in touch with the monitoring group at Environment and Climate Change Canada, and asked to see if I could help with monitoring and filling in some of these gaps. And they're particularly interested in some of the major tributaries draining the watershed on the Canadian side of the border, as well as understanding more about atmospheric deposition. Was that an important contributor to nutrient inputs to the lake? And this image on the right, I took from the TMDL report. So I just want to get rid of my face from the screen. This image over on the right shows the entire basin, US and Canada, outlined in black, and everything above the white line is in Canada. So you can see that there's more land area draining into the lake on the Canadian side. You can also kind of see from this Google Earth image some of the complexity in land cover and in superficial geology is evident in this image. And you can see here, can you see my cursor? Anybody? Kelly, I don't know.
Yeah, I can, Kathy.
Yeah, okay, perfect. Okay, so my cursor is kind of drawing a little line here within the Canadian part of the basin, sort of identifying this relatively small area here, which I'm going to sort of point out is a bit of a hot spot for nutrient losses within the Canadian portion of the basin. We call this the Agassiz clay plain. And you can see that there's a notable absence of lakes, right? We see a lot of dark pixels in the northern part of the watershed or in eastern part of the watershed. This is all Precambrian shield bedrock, imperfectly drained. A lot of the water that falls on the watershed collects in lakes. So absolutely littered with lakes in the Precambrian shield part of the basin. Whereas down here, which is where all the agriculture is, actually thicker soils and generally fine textured soils, quite a different geology, different soils, and also different land cover and different pressures. So it's a big watershed. As I said, there's no major cities in here. Thunder Bay is way over here somewhere. And so this presents some challenges. And so when you want to monitor something so big as this, you need to kind of come up with some clever strategies. It's also important to note the drainage patterns within this lake. Again, I stole this from the TMDL report. Thank you, MPCA, for all of the nice maps. The drainage patterns are also really important to be aware of when you're going out to try and monitor a gigantic basin like this. And so in particular, I want to draw your attention to these three major sort of sub-components or sub-catchments of the mega watershed. So the pink area here is primarily Precambrian shield geology. It really dominates the entire landscape, the entire watershed. And what's important about this pink area is that it ultimately drains into Rainy Lake, right? So it doesn't drain directly into Lake of the Woods. It doesn't drain directly into the Rainy River, which are sort of, we know, are sort of our problem areas. It drains into Rainy Lake. And then the Rainy Lake here exits into the Rainy River and snakes along into Lake of the Woods, right? And the Rainy River forms part of is the international boundary in this area. So this very large area that drains most of the Canadian Shield actually drains into Rainy Lake first and then exits via the Rainy River to the Lake of the Woods. The second largest area is this green area. Most of it falls within the US and in Minnesota. This little green area is that area I was showing you just in the previous slide on that Google Earth slide. This is primarily sort of that Agassiz clay plain, different soils, more agriculture going on. So this green area actually drains into the Rainy River and then it goes into Lake of the Woods. And there are a number of major, relatively major Canadian rivers that drain the green area into the Rainy River that we have been monitoring. So Sturgeon River, Everett Creek, Lavalley River, and then the Pinewood River that Environment Canada has been monitoring. I'll show you some data from those. And then probably many people from the NPCA, or certainly the American side, will be familiar with the Little Fork and Big Fork River, much larger watersheds that also drain into the Rainy River. And then lastly, there's this yellow area, excuse me, which is the direct lake shed. So these rivers draining this yellow area of land will drain directly into the lake. And we've been monitoring 3 rivers within the yellow part of the watershed. Berry Creek up in the north, it drains sort of Precambrian shield landscape. And then Little Grassy River and McGinnis Creek down in the south end drain Agassiz clay plain land cover, again, directly entering the lake. Okay, so when we were trying to design our study, we're cognizant of these drainage patterns and try to capture those within our study design. And so I received some funding from Environment Canada in 2018, and I was able to hire a field technician, a full-time field technician, was Wes Greenwood, now Andy Williams, buy a field truck, some field equipment, et cetera. But despite that budget, I still could not necessarily sample every river at the frequency that I wanted to. So we really had to be strategic in our sampling design. And so what we try to do is divide the watershed up into its 2 major geological zones, and then identify watersheds within each zone that we considered representative of the land cover within each zone. Okay, so we split it into 2 geological zones, Canadian Shield and Agassiz clay plain. And then within each, we identified representative rivers. And we did that using GIS, basically a very hurried GIS analysis that occurred in early 2018, preceded our initial sampling in spring of 2018. So here are the watersheds that we ended up selecting. So Berry Creek, Trout River, Seine, Atikokan, and Pickerel in the Shield zone, and then Little Grassy, McGinnis, Sturgeon, Lavalley were our rivers in the Agassiz zone, and Everett, sorry, And then the pine wood was being monitored by Environment Canada, and we collaborated with them on this project as well. And we also try to capture the end members of land use within each zone. So in the Canadian Shield, it's primarily forested, but half of the forest cover is coniferous. And the primary form of disturbance within the Canadian Shield zone is harvesting, and mostly clear-cut harvesting because it's a coniferous canopy. And so by end members, I mean, we're trying to capture sort of the natural end member, so, you know, untouched forest in the Canadian Shield zone, to the most disturbed end member, which in the Canadian Shield would be the most harvested kind of catchment that we could find. Likewise, in the Agassiz clay plain area, we tried to capture both natural, sort of untouched, relatively undisturbed watersheds in our sampling design, as well as the more agriculturally developed watersheds. So we had this sort of span the spectrum of disturbance and land cover within each zone. Okay. So here's just a few of those. I'll just show these fairly quickly. Some of the maps and the sort of the GIS analysis that informed the division of the watershed. And here you can see a superficial geology map of the Canadian portion of the basin that shows really clearly just how different the Shield zone is from the Agassiz zone. So in the Shield, we have lots of bedrock, right? So this is igneous and metamorphic rock that was scraped clean by the last glacial intrusion, leaving exposed this relatively slow weathering, phosphorus poor geology. And so the soils that developed over the last 14,000 years on the Shield area anyways are relatively thin, fairly coarse texture, dominated more sandy material, and low in phosphorus, naturally kind of low in nutrient content. In contrast, glacial Lake Agassi, so a lake that existed sort of during the height of glacial melting, which covered a lot of this southern area, deposited lots of fine textured materials, clays and silts, little tiny particles in this area, built up a much thicker sort of soil base. It's more poorly drained and lots of wetlands. And we can see down here, you know, we got a lot of till, also glacial fluvial, glacial lacustrine. So this is material deposited in ancient glacial rivers and lakes respectively. It's a very different superficial geology, which in turn determines the type of vegetation that develops in those areas, the type of human development that may occur. And here's the land cover layer. This is from Olmenson, 2015, also used by the MPCA. We tried to, I should mention, we try to be as consistent as possible across the border. Like I've tried my best to sort of collaborate with other researchers in this basin so that we can use, you know, the same tools, the same methods, and same sources of data in some cases too. So this is one of the examples of a land cover layer that we are using that the MPCA used as well to try and normalize our analyses. And this again shows, so if you follow my cursor, excuse me, this is the Agassiz zone. And we can see a lot more yellow. There's a lot of gray as well, or bluey gray. So a lot of wetlands in this Agassiz zone, but a lot of yellow. And if you steer closely, there's a fair bit of brown as well. So that's agriculture, row crops and small grains in brown, and then hay and pasture in yellow. So clearly all the agriculture that's going on in this basin is really concentrated in this sort of southwestern section. In contrast, we've got loads of lakes across the Shield. We've got a lot more forest, right? Coniferous and darker green, deciduous and sort of middle green. And then note also the extent of sort of this lime green sparse forest, right? So again, the legacy of clear-cut harvesting, there's a more regenerating forest within the Shield part of the basin. Okay, so again, we tried to capture the gradient of land cover, so disturbed to undisturbed, disturbed being more clear-cut harvested on the Shield, undisturbed being forest. In the Agassi basin, the disturbed was more agriculturally developed, and the less disturbed was really more wetland influenced. And this map, or data in this map, were provided to us by the Ministry of Natural Resources, showing sort of average harvest areas that have been harvested over the last 25 years or so. You can see most of these little red pixels are located within the Canadian Shield zone of the watershed. Not surprising, that's where all the forest is. And again, you can see with our watersheds, you know, we did a reasonably good job of capturing sort of the end members of disturbance within the basin. And so we see Trout River, lots of little red pixels in the Trout River. It's our most harvested watershed that we monitored. Saine's got a fair bit too. Whereas Pickerel River down here, almost untouched. It's very, very light, maybe less than 1% total harvesting, and Everett Creek's relatively low as well. So in addition to trying to capture sort of the gradients or members of land cover, we also had to adopt a bit of a stratified monitoring approach. Ideally, we would sample every river at least weekly and try to target storm events. We know from previous research that phosphorus loves particles. Phosphorus tends to bind to little tiny particles like clay and silt-sized particles. And so phosphorus moves with those particles. So any type of rain event or snowmelt event that mobilizes particulate matter off of the landscape, so those sort of erodible or erosive type events, will move with it phosphorus. So if you want to measure phosphorus, you've got to do a pretty good job of capturing those events. And those events can be very fleeting, right? You can have a big rain event, it comes, it goes, and there's a stream responds, and it's done. And so you have to storm chase. And again, given the size of this watershed, right? There's the scale down here on the right. And I have a field crew of 1. He can't storm chase this whole watershed. It's just practically impossible. And so what we tried to do is use a variety of approaches. So one, we used automated water samplers. So at the red dots at some of our rivers, we installed basically these little R2D2 units, little automated water samplers, have a whole bunch of little bottles inside them. You can program them to sample as often as you want, once a day, twice a day, 10 times a day if you like. You're limited by the number of bottles in the unit and how frequently you can go and collect samples from the unit. So those units basically are little automated water samplers helping us to do our job of storm sampling. So that was one approach. Second approach is Andy or Wes, they did drive around this watershed on a regular basis every few weeks to collect all the bottles from the ISCO samplers, but also to grab samples from what we call our synoptic sites, which are all in the blue. So those sites were sampled a little less frequently. However, we were lucky to collaborate with some great people living in the watershed as well as working in the watershed. And these, I call them lay samplers, have greatly enriched our sampling frequency. So specifically, there's some fantastic landowners in Everett Creek and at McGinnis Creek who basically, not only did they allow us to install equipment on their properties, but they also very kindly offered to sample the river when it was storming or when it was raining. So Andy or Wes would give them some bottles and ask them to go out and sample when it was raining. They did so, put the sample bottles in their fridge, and then Andy would go around and collect them when there were samples to collect. So that allowed us to capture a lot of storms that we just simply would not have been able to do on our own. We've also been collaborating with folks in the Experimental Lakes area, IISD, so Mike Patterson, Scott Higgins have been really fantastic about sharing data that's collected up there. So that's allowed us to capture the northern part of the basin without having to go there physically ourselves. And there's been some Parks Canada folks and M&R folks who have also helped us at sites like at Quetico, for example, with some storm chasing as well. So that's really, a little help from your friends kind of thing has really helped us to blitz this gigantic watershed. I'm not going to talk about deposition in this talk, but I just want to also point out that we also have been monitoring atmospheric deposition at the purple squares. And we've also done some snow surveys to try and capture what's going on in the winter. The winter is historically an understudied season, yet it's very, very important, especially for setting up the spring melt. And so understanding nutrient dynamics, inputs, and the pool available for subsequent flushing during the spring melt is really important, an important element of our sampling design. And so we have done year-round sampling, not only of the tributaries, but also of atmospheric deposition to kind of inform our analysis of tributary and deposition sources. Okay, so a few photos of monitoring in action. Excuse me. So here's this ISCO up here, the top left photo. This is open, so the top is over here on the left. This has got the brains, right? It's the part that you program. Over here, you can see all the glass bottles inside. And so we would program the ISCOs to take a sample every single day. And then when Andy would come and retrieve all the bottles, he could download the data and we could figure out, which samples were taken on what, part of the hydrograph. So we could target which samples we wanted to analyze chemically. Down here is Andy taking a stream gauging measurement. I didn't mention this, but in addition to monitoring water quality or the concentrations of nutrients in water, You also want to measure the volume of water flowing through these tributaries, right? Because you multiply concentration times volume to get mass. So in order to get good budget estimates, we need to not only quantify concentrations and do that storm targeted sampling that I mentioned, we also need to really accurately quantify the volume of water moving through these tributaries. So we've spent a lot of time and effort on this part of the budget calculation as well, instrumented these streams. There wasn't very much flow data available within the Canadian portion of the basin, so we've really worked to try and enhance that portion of the monitoring effort. Excuse me. Over here is Wes taking an in situ measurement. So we also measured in situ turbidity, took samples for total suspended sediments. There's Wes monitoring through the ice. And here's some deposition collectors over on the left. Again, demonstrating sort of the year-round multi-season approach. So on the left, far left, you can see sampling during the growing season. The collector is topped with a funnel to sort of limit evaporative losses of water. And there's a little filter inside to sort of block leaves and things like that from getting into the collector and sort of influencing the chemistry of the water in that bucket. And then over here on the right, you see the same bucket basically without the top, and it's open to capture snow. So here's just a few pictures to show you what those watersheds look like, or what the rivers look like, I should say. So these two rivers are in the Agassi zone. So this is our sort of more, flatter topography, thicker soils, finer textured soils, more agricultural development. So Sturgeon River is kind of at the sort of more developed end of the spectrum. McGinnis Creek is at the least developed end of the spectrum, so mostly natural. And you can see what those rivers look like. And if you look carefully through the shrubbery, you can see the ISCO sampler on the right side of the Sturgeon River riverbed. And this is again two rivers where we had really fantastic landowners helping us out, letting us install equipment on their property. And the property owner at Sturgeon, even, he would like whippersnip around the ISCO to keep down ticks. Ticks are horrible in the southern part of the basin. So really appreciate those folks and their efforts to help us out. So going north onto the Shield, these are two sort of representative rivers. The Trout River on the left in the Shield zone, this is our most clear-cut harvested site. And on the right, Pickerel River, our most natural Shield site, just to give you a sense of what they look like. Okay, let's get into some results. This is what you're waiting for. So the first image I want to show you, or graph I want to show you, is the full record of water quality and quantity that we have for Sturgeon River, which is, again, that M member of sort of disturbed or agriculturally developed watershed in the Agassi zone. Sturgeon River is one of my favorite rivers because it's the most clean patterns of water quality. You can give me this figure and not label it with a name, and I can recognize Sturgeon River because it's just such a predictable pattern of water quality, which is really great for monitoring, because it means that this river, we can use this river to sort of predict what's going on at other rivers. So a few things I want you to take note of in this graph. So first of all, the blue line is showing river flow or runoff in units of depth, millimeters per day. So what you can see, so we started monitoring in April of 2018, and we continue to monitor actually even through COVID. We, mainly due to Andy's determination to keep going, and again, the Ministry of Environment and help from those guys to keep analyzing our samples. Could not have done it without them, but we managed to keep going through COVID. So I do have data beyond this date. I have it through 2021, but this is what's worked up so far. So the blue line is showing stream flow, and we express it in units of millimeters or depth for a couple of reasons. One is because it allows us to say, or to compare a runoff response with the amount of precipitation coming into a watershed. So for example, it rained for we have an 8 millimeter rain event. We're quite comfortable with units of millimeters when we're talking about precipitation. Well, if I want to say, well, how much did the stream respond to that 8 millimeter input of precipitation? It's helpful if I have my units of runoff in millimeters as well. So I can say, if it responded by 1 millimeter to that 8 millimeter runoff or precipitation event, I'd say, modest response. If on the other hand, 7 millimeters of runoff was generated from an 8 millimeter precipitation input, that would be a massive response, right? So it helps us to compare precipitation inputs with outputs by using runoff in millimeters per unit time. Secondly, it allows us to compare watersheds with other watersheds of different size. Sturgeon River is about 170 square kilometers. And I'm going to show you some other rivers that are smaller or larger than this. And so if we express everything in units of millimeters, we're standardizing for area, differences in area. I couldn't do that if I presented data in units of volume, meter cube, for example. Okay, so that's the blue line. So another thing to consider is sort of the seasonality in the blue line. So the blue line is pretty, what you should notice is that it's a lot of ups and downs, right? And when it's down, right? So this blue line is pretty much flat line in that first summer that we monitored. It was sluggish, like it was hard to tell which way the river was moving, if at all. It was basically sort of a pea soup at this point in time. Other times it's really, really high, right? And we look at this event here. This was, if anybody who lives in the basin, the summer of 2019, a lot of rain, flooding everywhere in the southern part of the basin. So this is the runoff response in Sturgeon River, a lot of flow. So what we can see from this, it's drought to deluge, basically. It's very low or it can be very high. So overall, we see a very flashy hydrology at Sturgeon River and at other rivers within the Agassi zone. And this is very important to note because it means if you want to capture those events that I said are so important for phosphorus export, you need to capture those events. And they come up and they go down relatively quickly for such a large watersheds. Furthermore, the range in phosphorus concentrations is extreme, right? So if we look at this graph, so here is phosphorus on the left side of the axis, the left y-axis. We can see that the lowest concentrations of phosphorus ever measured in this river for our 2.5 year record that's shown here, it's just a little below 50, right? So maybe this is 45 micrograms per liter. The highest were up to 400 micrograms per liter, right? So it's like a 10-fold difference in phosphorus concentration. And not only is it an extreme range, but those changes occur very rapidly. So here, the very first measurements Wes took, 400 micrograms per liter at the height of spring melt. This is the height of spring melt right here. This is the peak snow melt. Maybe a week, 2 weeks later, it's down below 50 micrograms per liter, right? So here's another example, right? Peak of 255 micrograms per liter or so in the summer of 2018, and it drops by the fall of 2018 to 40 micrograms per liter. So extreme range in phosphorus concentration. So I would call this a flashy, not only flashy hydrology, but flashy phosphorus regime as well. And this really explains why frequent monitoring is essential in the Agassi zone. You can't monitor this in monthly. You can't go there once a month and expect to capture this and understand, the drivers of phosphorus release in this landscape without this kind of high frequency data. The other thing I want to mention from this graph, there's lots of things I can mention in this graph, but just one other little thing to mention in this graph, which is important, is that how phosphorus concentrations seem to respond to changes in hydrology. Right, so here we have a very, very dry summer. The summer of 2018 was extremely dry. Phosphorus concentrations peaked. They were really, really high, above 250 micrograms per liter in the summer when flow was at a minimum. Then they decline through the fall runoff. But we have also other instances when phosphorus concentrations are very high when flow is high. So normally we think of, or what I said to you before, is that phosphorus moves with events, right? So therefore we'd expect phosphorus to increase when flow increases. And we do see that sometimes, but we also see the opposite. So we see phosphorus doing crazy things, high under low flow, high under high flow. And that is an important observation because it helps us to understand what is controlling phosphorus. Is it just erosion? No, it's not just erosion. There's more going on in this watershed. Last thing to pick out from this graph, I'm just showing the Minnesota Northern River guideline, 50 micrograms per liter. 30 micrograms per liter is the Ontario Provincial Water Quality Objective for phosphorus applied in Southern Ontario. So this is high, right? These are high concentrations. These, by guidelines, those guidelines are based on trying to prevent eutrophic conditions. So these are clearly, or this is clearly a very eutrophic river, very high, consistently high phosphorus concentrations. Okay, there was one more thing I want to point out from this graph. Last thing I want to point out from this graph is that we have, and this is relevant to some of my later slides, I'm going to roll these data up and to present annual totals or annual averages. When I'm talking about annual averages, I'm talking about the hydrologic year, not the calendar year. So these data represent 2 hydrologic years, and our hydrologic year is October 1st to September 30th. So when I talk about the 2018-19 hydrologic year, I'm talking about October 1st, 2018 to September 30th, 2019. So I just want to keep that in mind when we talk about annual budgets later on. Okay, so I was trying to make the point that we're trying to, our sampling design was meant to capture end members of land cover, disturbance, but we're also trying to identify representative sites to guide future monitoring. This is a big watershed. We can't monitor everything. As a part of our purview was to help, feed back to Environment Canada and to Ministry of Environment to say, here, these are what we recommend in terms of which sites are the best ones to sample, how frequently to sample. So it's important for us to compare, Sturgeon River with Pinewood, with Everett Creek, and see, are we actually doing can we actually use one watershed as sort of a sentinel watershed, as a representative indicator watershed of these other ones? Can we kind of not sample them quite as frequently? And the answer is yes. The answer is yes in both the Agassi zone and in the Cambrian Shield zone. And this helps to justify that decision. So Pinewood River on the left was monitored by Environment and Climate Change Canada using an ISCO as well, but a different ISCO from mine, sent to a different lab. And what I find so encouraging about this figure is that even though it was a different sampling design to a certain extent and different laboratory methods, we still got very, very similar results. So that's also encouraging. It gives me some confidence in our numbers. And if you remember in that first summer, the dry summer, here you see it at Pinewood as well. Remember, phosphorus concentrations peaked, right? They went up a lot. It didn't go quite as high in Pinewood, but 200 micrograms per liter relative to the 250 in Sturgeon. Same sort of pattern occurred. Flow tanked, phosphorus peaked. Then phosphorus declined as the fall rains occur, right? So phosphorus declined here. We also found some increases under ice. We saw that at Sturgeon River as well. And here's their second year. So we see a very similar seasonal pattern at Pinewood as we saw at Sturgeon. So that's somewhat reassuring. Likewise at Everett Creek on the right. This one we didn't have an ISCO, but I love this figure because it shows just how fantastic our lay sampler, our help, the lady who, I don't want to name names, but she's fantastic support of our project. She went out and sampled Everett Creek when it was raining. And you can see by the number of points on this graph that she did a pretty darn good job. She's almost like a human ISCO. And so we, again, see a very similar pattern of an increase in phosphorus, even higher than at Sturgeon or at Pinewood, topping out at 450 micrograms per liter. So we see very high concentrations in that summer low, decrease in the autumn wet up, but again, we see some really high concentrations at high flow as well, right? So it's a dual response. Low flow generates high concentrations, high flow generates high concentrations. More than one thing going on. It's not, we would be oversimplifying just to say this was an erosion story. It's just, particulate matter being shunted out during rain events. It's not, that doesn't explain what's going on in the summer when we have these very high concentrations. So take home, it's not necessary to monitor every river. So that's kind of the good news. One other little thing I want to point out here, I mentioned, or maybe I forgot to mention at the very beginning, most studies have focused on phosphorus, and we're measuring nitrogen as well, because it is not just, growing recognition that, a more holistic approach to nutrient budget studies is warranted. You get more insight into drivers of algal blooms if you look at not just phosphorus, but also nitrogen, carbon, other elements as well. So we are measuring nitrogen as well, and nitrogen levels are much higher at Everett Creek, incidentally, where we have a lot more agriculture. The other thing to point out is these very high concentrations that occur in the summer during low flow. A lot of people will say to me, well, why do you care? High concentrations, low flow means low mass. Who cares, right? It's just a small amount of kilograms of phosphorus coming out into the Rainy River and to the Lake of the Woods. Well, I would suggest we should care because algae care about concentrations. They don't care about the mass of phosphorus, they care about concentrations. And so when you have these really, really high concentrations during these dry summer conditions, perhaps this is setting the scene for those fall blooms that occur. Maybe, we don't know, but I would say it is a bad idea to underestimate the importance of very high concentrations during low flow and just focusing completely on mass. Understanding and looking at concentrations is also important when understanding drivers of nutrient blooms or algal blooms. Okay, now I'm flipping north and east, I guess, onto the Shield, and we see a very, very, very different flow regime, as well as a different pattern of phosphorus dynamics. So this is Pickerel River, which is our sort of cleanest, if you like, or most natural, undisturbed river that we monitor on the Canadian Shield. Now note the y-axis here is the same scale that I used for presenting runoff at Sturgeon, Pinewood, Everett Creek. Notice that we don't see as many high peaks in flow as we saw at those Agassiz rivers. And this is the same across all the shield basins that I'll show you. Total runoff is just far more muted. It's more it's not sluggish is not necessarily the right word, but it's certainly more buffered and not as responsive to rain events. So you can have the same rain events occurring on the shield zone, and they don't generate as rapid or as large of a response in runoff as you see in the Agassi zone, right? So the shield rivers just seem to be more well buffered. And we think that's because of all the lakes, all those lakes that litter the Canadian shield landscape. They're providing storage, right? So it's sort of fill and spill kind of hydrology. So all those, rain events can occur and they'll generate some runoff, but you have all these big lakes that are sort of helping to swallow up a little bit of that response and sort of mute the hydrologic, the flashiness. So these rivers are nowhere near as flashy. And from a monitoring perspective, that's kind of a good thing because it means you don't necessarily have to be out there running after those events and getting out there in your truck and driving, madly to capture the storms. You don't need the ISCOs to sample the Canadian shield rivers. A more infrequent sampling regime is adequate on the shield, is sort of my take-home message. And likewise, we have a much smaller range in total phosphorus concentrations. Note the y-axis on the left of this figure. On the other graphs, I was showing you, 400, 500 micrograms per liter, right? So here's the maximum is 20. And the maximum measured in this river, one measurement above 14 micrograms per liter. The vast majority of these little black dots are kind of falling between, 5 and 10 micrograms per liter, really pretty low. So this is kind of oligotrophic condition. So much lower phosphorus, maybe a 3-fold range between year, compared with more like a 10-fold range in the Agassiz zone. So much less jumpy kind of hydrology or phosphorus regime on the shield zone. And this is again, a natural stream. Here's our most disturbed river that we monitor. So this is the Trout River, 30% clear cut harvested. And I should note that this was not an intentional part of our study design, but Wes sort of noticed after he started monitoring that there was some cutting going on immediately upstream of our sampling location. We were a little worried about that. We thought, oh, we're gonna be run over by a logger or something, but it actually worked out. I think we learned some interesting things from being immediately downstream of a cutting operation, because we actually captured some of the sort of that brief pulse of nutrients that might come out from cutting. So what you can see here at Trout River, we had some pretty high concentrations for the shield zone. These are the highest concentrations I've ever seen, but they were relatively brief, right? So we had higher phosphorus concentrations in our first year in 2018, but by my green blob's in the wrong spot, but by 2019, we're pretty low, right? So all these dots are kind of hovering in the same general range as what you saw at Pickerel River, sort of in the 5 to maybe, a few going above 10 micrograms per liter, for the most part, 5 to 10 micrograms per liter. So it seemed to kind of recover, if you like, relatively quickly, suggesting that perhaps there was a brief sort of harvesting effect that we captured, but after that was done, the phosphorus concentrations kind of returned to sort of background or normal concentrations for the zone. And again, much less flashy hydrology. The other thing to note here is you basically have 2 peaks year after year after year. So there's the spring melt peak shown right by my cursor here, and then what we call the autumn flush. This is the autumn flush. This is the crazy peak that you saw in Sturgeon River when I said there was like huge amount of rain and really high runoff response. In the Agassi zone, there was higher runoff on the shield as well, but nowhere near as pronounced as what you saw in the Agassi zone. It exceeded the spring melt pulse, certainly, but it was nowhere near as large as it could have been without all those lakes that helped to kind of buffer the hydrologic response. Okay, so now if I put all of that data together from all of the rivers that we've been monitoring on the shield and on the Agassi zone, and this is just all the raw observations of total phosphorus concentration. And so these are box whisker plots, which help to kind of summarize all those individual observations. And so these box whiskers, the line in the middle is the median concentration, and the little X is the average concentration, or the arithmetic mean. And I've shown both the shield on the left and the Agassi on the right, along a gradient of disturbance. So within the shield zone, this is Trout River, the most disturbed. The least disturbed is Pickerel, and in the Agassi zone, my least disturbed is McGinnis Creek. My most developed is Everett Creek. And you can see a general increase in concentration with increase in disturbance, but it's not as pronounced as you might expect. In fact, even Little Grassy River here in blue, which has very little agricultural development, less than 10%. Concentrations are not dissimilar. They're certainly overlap with Pinewood and with Lavallee River, and even with Sturgeon to a certain extent. So suggests that while agriculture and disturbance associated with agriculture probably is responsible for some of those high concentrations that we're measuring, it's not the entire story. That natural background concentrations are rather high in this landscape. It's a naturally erodible landscape, and probably quite sensitive, though not probably, it is quite sensitive to disturbance. Okay, so this really summarizes everything together. And if I put it back onto that mega watershed map, so the pink zone, which dominates the Canadian portion of the basin, represents 60% of the entire Lake of the Woods watershed is in Canada, and probably about 85% of it is in the shield zone in Canada. Phosphorus concentration is very low, so 5 to 15 micrograms per liter. And again, these ultimately drain into Rainy Lake, which then exits via the Rainy River into Lake of the Woods. So my take home from this is that the shield zone is pretty low, pretty oligotrophic. Harvesting appears to cause sort of an ephemeral pulse of phosphorus that may be of concern to downstream lakes within the immediate term. But generally speaking, in terms of management and focus for the future, in terms of driving algal blooms, the Precambrian shield is not the area that we really need to worry about. In contrast, down here in our green zone and our yellow zone, we see much higher concentrations. This is all from the Agassi zone. So the Agassi zone is our hotspot. So here we have, when I just summarize the volume weighted concentrations, Little Grassy, McGinnis, direct lake shed rivers, 40 to 75 micrograms per liter, exceeding that 30 micrograms per liter provincial water quality objective for Ontario, almost exceeding the 50 micrograms per liter northern water objective in Minnesota. And likewise, all the Rainy River tribs, Everett, Sturgeon, Pinewood, Lavallee, much higher, 75 to 150 micrograms per liter. And just for reference, for the MPCA folks who might be on this call, these I just took directly from the TMDL, Big Fork, Little Fork, 50, 70, 50 to 80 micrograms per liter. So we're kind of in the same ballpark, although generally for the 2 years of record that I have anyways, we had some higher concentrations at the Canadian tributaries. So this is really our hotspot for focusing management from these data anyway. Lastly, I tried to scale these things up. So that was concentrations, which the algae care about, but when you're doing a nutrient budget and you're looking at, trying to identify important sources and the contribution from those sources, you need to calculate export in kilograms. So here I've shown total phosphorus export in kilogram per kilometer squared. So I'm normalizing by catchment area. Again, we monitored over 2 hydrologic years, 2018-19 and 2019-20. The first year was relatively wet. We had about 400 millimeters of rain occur in the summer of 2018. The climate normal, 30-year climate normal is 250. So a lot of that wetness was to do with that crazy rain event or runoff event that you saw in summer of 2018. In contrast, the 2019-20 was relatively dry, below the long-term climate normal. So this is good in that we captured, I think, a little bit of, not probably the full range of hydrologic variability. We did a decent job of capturing, a low year and a high year. And that gives us some sense of, whether we need to do some more monitoring, what the response might be of these watersheds to future climate change. It gives a little bit of insight into sort of the sensitivity of these basins to future change. And so you can see in the blue line, we have all of our wet year export estimates. So generally pretty high across the board. especially when you consider the dry year right beside it. So in orange, we see the dry year export. So a lot higher. For example, at Everett, it was 3 times more export in the wet year compared to the dry year. The Shield River or Shield tributaries, much lower export, not surprising, given the very low concentrations. Similar differences year to year, but overall, the height of those bars is pretty darn small. And really, again, we're seeing it's the Agassi zone that's kind of exporting the greatest amount per unit area. I also added on TMDL numbers. So I took these from the TMDL report. So some of the Canadian rivers were included in the TMDL report. They were modeled from some past monitoring data or chemistry data and modeled flow. And we can see that in the dry year, we actually do, like the TMDL numbers actually match fairly well. Not so much like Little Grassy, but for McGinnis, sorry, not McGinnis, Pinewood, Lavalley, and Sturgeon, not too bad. But for the wet year, we have a lot more export. And the last thing I've added here, the Little Fork and Big Fork, just to show you that the Canadian tributaries are exporting as much or more per unit area compared with the Little Fork and Big Fork. Sorry. Thanks. Okay, so some considerations for future monitoring. As I mentioned, these rivers in the Agassi zone are clearly very responsive to hydrologic change, to events. And I love these 2 photos because they really show that very clearly. So this is Everett Creek, which is our most agriculturally developed watershed. Summer of 2018, we can see it's flowing away. And then this past summer, there's Eric standing on a dry riverbed. And I remember very clearly the first year I was visiting the watershed, and Wes and I were standing up here on this road above these culverts. And I was like, what is going on with this? I just couldn't understand why they had 3 culverts on top of each other. I've never seen such a crazy sort of storm drainage design. And we were talking fairly loudly, maybe, I don't know, but it just so happened that a landowner heard us and decided to come over and sort of talk to us about the culverts. And he said, well, you should have seen it, and he started rhyming off dates when the flow actually went over this road and justified the need for, multiple culverts. So I think this is a great example of sort of the drought to deluge potential for Agassi's river watersheds, right? So if climate, weather becomes even more variable, we're going to see, certainly in the Agassi zone, we can expect to see a highly variable hydrologic response and highly variable phosphorus response. So more years of data, in my opinion, are needed to capture that variability. We've caught a dry and a wet year, and now when we start to work up the summer data from this past year, we've got an even drier year to look at. But really, I think the Agassi zone data show you that it's a bit misleading to provide an average, right? We need to kind of see what are the extremes, the highs and the lows. Crazy culverts seen in the background. In addition, their land use change is ongoing in the southern part of the basin. As I mentioned before, Canada is not exactly well endowed with land, good soils, good climate for growing crops. And so of course there's an interest in expanding agriculture. So climate warming is not a bad thing for agriculture. However, it could have unintended consequences for water quality. And this is just something I want to put on the radar for future monitoring efforts, for future management efforts, is that there's a lot of change going on. I showed you, the 2015 Olmanson layer shows a lot of hay and pasture. Well, a lot of that's shifting. We're having a conversion from hay and pasture towards more intensive row cropping. And that can be corn and soybean, other row crops. But corn is a particular concern for leaching nitrate, something we need to be aware of. And tile drainage is going on in gusto in this watershed. And there's an Ontario government resource, funding source that helps farmers finance tile drainage. And actually, this is just from September 17th headline from Northern Ontario Business showing that $1.3 million investment for tile drainage going on in the Rainy River area. And it's also going to support the establishment of a new business in, I think it was in Fort Francis. So a new, so a Southern Ontario tile drainage company is going to be building a new site in Fort Francis. So there will be, likely more tile drainage installation going on. And so tiles, if you're not familiar with tiles, so tiles are like basically perforated plastic pipes that get inserted beneath fields, maybe 80 centimeters to 100 centimeters deep. And you have lines in them, maybe 10 meters apart. And those lines go under the soil, and then they connect to a header tile. And then that tile basically captures soil water that drains below the rooting zone, and it discharges it rather effectively into ditches or rivers directly. And so it's a good thing for crops, because it helps to drain those seasonally saturated soils, allows you to get your equipment out there earlier and harvest a little later, extends the growing season basically. And it might actually be a good news for phosphorus to a certain extent, because what it does is it helps to encourage sort of vertical drainage of water. So when you have those big flow events, those rain events that would normally cause lateral or overland flow, it might actually increase storage in the soil and allow more water to penetrate the soil, mitigating a little bit of that erosive action of those events. So might help to decrease total phosphorus in the particulate form losses. However, tiles are known to augment dissolved nutrient losses, which is the more bioavailable fraction of most nutrients. So nitrate losses tend to be higher in tile drainage, and dissolved phosphorus concentrations can also be higher in tile drainage. So something to be cognizant of. There's really no there's no limits around putting in tile drainage. You don't have to apply, do an environmental impact assessment or anything like that. So it's something we should be aware of. And as I said, there's a fair bit of funding to encourage this in Northern Ontario. That's all I wanted to talk about for today. I want to again acknowledge all the folks who have helped us. I should also mention a shout out to Dorset Environmental Science Center, who's done all of the chemical analyses for us, including through COVID. So Andy was still sampling through COVID, and they were opening up their lab to accept our samples. So could not have amassed these data sets without their help. And happy to take questions.
Thanks, Kathy. That was great. One of the things that jumped out at me, but I'm not surprised, was the collaboration you got from other researchers and also members of the public. That seems to be kind of a character trait in this watershed. So, it was just really neat to hear how that, helped you get places that you couldn't get otherwise, and in really important time periods too. So really good to hear.
Mm-hmm. Yeah, I'm really very thankful for their efforts over the years. Yeah. And also just acknowledge to Wes and Andy and their efforts for, not only do they have great field and technical skills out in the field capturing, events and etc., but also great people skills, right? To be able to, facilitate those connections and maintain those connections has been really important.
For sure. Okay, well, we have just a couple of minutes because we like to finish off right at 1. So if anyone, I don't see any questions in the Q&A or hands raised, but this is your chance to ask Dr. Eimers a question if you have any. Todd has raised his hand.
Well, I'll kick off the questions, and thank you very much, Dr. Eimers. The graphs you presented comparing the loads per kilometer squared between and among the rivers, I think was quite interesting. And that, of course, gives us sort of an aerial export load comparison. What would be the total loading comparison be like from those rivers?
Mm-hmm. Yeah, so that's the important note, I guess, is that it's kilograms per kilometer squared times kilometer squared gives you total kilograms. So size of the watershed also matters. So big fork, little fork, of course, much larger watersheds are going to export a lot more total kilograms. But I guess my point is that that's important, right? When you're doing a mass budget, you want to take into consideration the total amount of nutrients coming from each sub watershed. But when you're trying to focus management efforts, it's helpful to express it per unit area, because Everett Creek is exporting more per unit area than the Little Fork River, for example. Yet the Little Fork River is seen as sort of being this nasty, source of phosphorus. So I think it's important to, in terms of management, if you were trying to identify sites to focus, best management practices. You know, looking at kilograms per kilometer squared is helpful.
Good, thank you. We are open for other questions. Oh, we have a hand raised, Bill Taylor. Bill, if you could unmute, please.
Hi, Kathy. I was curious whether you have any information on the speciation of phosphorus between the watershed types and particulate and dissolved or anything like that?
Yeah, so we just did total dissolved and then total phosphorus. So we do, I suppose, have phosphate as well. I haven't looked at the phosphate as much because I'm not super, you don't necessarily trust that as much, but the total dissolved phosphorus concentrations, I was expecting to see greater differences across sites, but actually end up getting about 50%. It varies, right, between events and non-events, but it didn't differ quite as much as I was expecting. And you end up getting something like 30 to 75% dissolved, sort of hovering around a middle point of 50%. So yeah, it wasn't quite as illuminating as I was expecting.
Thank you.
Any follow-up on that, Bill?
No, I'm happy with that response.
Thanks.
And I can show you some of the results too, Bill, if you want another time.
Bill, your hand is still raised. Oh, you're gone. Okay. Okay, we're at the top of the hour. I don't want to cut off anyone that has a question. So one last opportunity to ask before we close out. Okay. Well, thank you everyone for joining. Just a reminder, if you have a friend or colleague who missed today's presentation, in a couple of days, the recording will be up on our foundation website. And we will do that for all of the presentations this fall. Special thanks to Dr. Eimers for enlightening us with really critical information for understanding this watershed. We appreciate your hard work and that of your technicians as well, and grateful to have you here in our basin and sharing your information. So thank you so much. And everyone have a wonderful day, and thank you for joining us. See you next week.